Hello, everybody. This is Nafrit Sekho, and I welcome you on this platform, The Layers of Life Sciences, where we are discussing different aspects of life sciences in quite depth and detail, layer by layer. The objective is not just to help you in your graduation, post graduation, and competitive exams in clearing CSIR net, but also to have a general conversation about the life processes occurring around us. We have started this journey with the discussion of the structures of the molecules of life, that is, the biomolecules. In the last 5 videos, we have discussed the structure of monosaccharides, the basic building blocks of one of the biomolecules, which is carbohydrates, in quite detail. Today, we are going to extend that discussion with the structures of the derivatives of these monosaccharides. Now, what are these derivatives? These derivatives are actually the molecules which are formed by some kind of modification in the basic structures of the monosaccharides. These monosaccharides can be aldoses or ketoses, which means it can be glucose, it can be fructose, it can be galactose, it can be xylulose, it can be anything. But whenever there is any kind of modifications in their structure, then the molecules with the resultant molecules which are formed are actually the derivatives of these monosaccharides, and they are called sugar derivatives. Now, uh, we need to discuss these structures in detail. Let's start with the first ones, which are called the amino sugars. So therefore, this is the most abundant and most commonly occurring monosaccharide, which is the D form of glucose, that is beta D glucose. So if we are taking this, you can see this, what is the general structure, that at each of the carbons, there is one hydroxyl and one hydrogen associated with it. Apart from this carbon number 5, where it has lost its hydroxyl group and it is associated with the hydrogen and the carbon number 6. Over here, the carbon number 6 is directed upwards. You can see, right? Hydrogen is directed downwards and CH2OH is directed upwards. Whenever CH2OH is directed upwards, it means it is the structure of D-glucose. If this CH2OH is mentioned over here, where H is mentioned right now, then which means that if CH2OH, the carbon number 6 is shown downwards, then it is always the L form of the glucose. But now, right now, it is directed upwards. It means it is the D-glucose. Now, we know that the code for the linear form of the glucose is 245, right? We know for that, what is this code? For that, you will have to refer to the video Biochemistry 2. It's very important to learn the codes of those linear structures. They are gonna help you a long way, be it the general structure of the carbohydrates, be it the metabolism of carbohydrates, be it when we will be doing our cellular biology or molecular biology. If you know the codes, none of the structures will be difficult for you. So this is 245. We know that 245 is the code for glucose from the video Biochemistry 2. If you have not watched it, watch it first, and then all of this which I'm talking right now in this video will make sense. So glucose is aldohexose, right? It means 6 carbons. Which means in the chain of 6 carbons, at carbon number 2, carbon number 4, and carbon number 5, the OH is directed towards right-hand side. The OH is directed towards right-hand side, while if that is true, that it means The only carbon, interstitial carbon, which is left is carbon number 3. On that, OH is directed on the left hand side. Now, as it is aldohexose, it means a carbonyl group is present on carbon number 1 along with other valency. Now, this another achiral carbon, which is at the other terminal end. Over here, it will be CH2 and OH, right? So over here, rest of the positions can be filled with H. Now, when this structure gets translated into the cyclic structure, there again we did a rule, right? If you will refer to video number 3, Biochemistry 3, there we converted the aldoses into the cyclic structures, right? So there we actually did that in whatever position in the linear structure, OH is on the right-hand side of the carbon skeleton. On the same positions in the cyclic structure, OH will be directed downwards. So over here at the position number 2, OH is on the right-hand side. So over here, this is carbon number 1, this is carbon number 2. So over here, OH is directed downwards because in the linear structure, it is on the right-hand side. Same on the carbon number 4 over here. OH is on the right-hand side. That's why OH in the cyclic structure is directed downwards. Now, about carbon number 5, we know that in the process of cyclization, when this hydroxyl group present on carbon number 5 attacks the carbon number 1, in that process, carbon number 5 loses its OH. Now, after this, carbon number 5 is not left with any OH. So this last position will not be considered for the cyclic structures. So for the cyclic structures, first two positions matter, which means that on the cyclic structure of glucose at position number 2 and 4, OH should be directed downwards. So over here, this is a pyranose ring, and in this pyranose ring at the carbon number 2 and 4, OH is directed downwards. That's why this is glucose. First point, because on the carbon number 5, CH2OH is directed upwards, that's why it is D-glucose. Third point, because on the carbon number 1, OH is directed upwards, that's why this is beta-D-glucose. So this is what we have actually done in detail in the last 5 videos, right? Now, this is one of the most common monosaccharides, which we will come across again and again throughout this series of life sciences. Now, the next one is another epimer of glucose, which is mannose. Now, for mannose, the code was 45, right? The code was 45, which means that on the carbon chain composed of 6 carbons, at carbon number 4 and 5, OH is directed on the right hand side of the carbon skeleton. Because over here on these two positions, OH is directed on the right hand side, that's why OH on the 4th position should be directing downwards, while because over here the 5th position in the process of cyclization loses its OH, so we cannot consider this position. So over here, so how mannose in the cyclic structure will look? All the OH apart from OH on the carbon number 4 will direct upwards. On carbon number 4, OH will direct downwards because its code is 4 for the cyclic structure. Then we come to the next epimer of glucose, which is galactose. So now, over here, we know that the code for galactose was 25, which means on the chain of carbons, which is formed by 6 carbons, 1, 2, 3, 4, 5, 6 carbons. On the position number 2 and position number 5, OH is directed towards right-hand side. But in the process of cyclization, this carbon number 5 will lose its OH. So we are left with left with the OH on the carbon number 2. So in galactose, just on the carbon number 2, OH will be directing downwards, while on all other carbons, all other interstitial carbons to be precise, OH will be directing upwards. So to conclude, in glucose, the hydroxyl group will direct downwards on carbon number 2 and 4. On mannose, it will direct downwards just on carbon number 4, while in galactose, the OH will direct downwards just on carbon number 2. So these are the basic, most abundant occurring or most abundant monosaccharides which we come across during our studies. Now, we actually want to talk about the derivatives of these monosaccharides. Here, all the carbons are associated either with the other carbons or with H and OH. So OH, that is the hydroxyl groups, and the H are their major substituents. Now, whenever this carbon gets associated with any other group apart from any other carbon or any other H or OH, then derivatives are formed, right? So let's see how. This is the glucose molecule, right? So this is the glucose molecule, and this is the hydroxyl group attached to the carbon number 2, right? When this hydroxyl group over here gets replaced by amine group over here, then OH gets replaced by this amine group, then the molecule, very important molecule, which is called beta-D-glucosamine is formed. It can be called glucosamine because over here it has been derived from beta-D-glucose, so that's why it is beta-D-glucosamine, right? It could be alpha-D-glucosamine if we would have done the derivation from alpha-D-glucose, right? But beta, as we know, beta is the most abundant form of glucose. That's why we are doing the beta form. Then same thing will happen when this OH, which is again present on carbon number 2 of mannose, again gets replaced by amine group, that is NH2, then monosaccharide is formed. Beta-D monosaccharide is formed. Similarly, when this OH on the carbon number 2 of galactose gets replaced by amine, that is a NH2 group, then galactosamine is formed. So these are the most common amino sugars. I have taken the examples of 3 monosaccharides, but this can happen to any kind of monosaccharide. It can happen to even fructose. Now, let's move forward. As we know that glucose is the most common monosaccharide, let's learn few of its derivatives, right? This is the glucose molecule. When OH gets replaced by NH2, glucosamine is formed. Now, other groups are equally prone to get replaced. So let's see how. So over here, when this H, which is the part of OH group of present on carbon number 6, just H of this hydroxyl group of carbon number 6 gets replaced by phosphate group, then very, very important derivative of glucose is formed, which is called glucose 6 phosphate. Then it is called glucose 6 phosphate. This is very important derivative. We will come across it when we will be doing the metabolism during glycolysis, the first step. So now, similarly, we have done this, although just to keep all the derivatives of sugar on at one place. I've redone it. When this hydroxyl at carbon number 2 is replaced by amine group, then beta D-glucosamine is formed. Now, this beta D-glucosamine has been formed. When again, in this beta D-glucosamine, we can see there is one N and there are two H's. When one of these H's gets replaced by acetyl group, what is acetyl group? I will explain. One of the H's at this position gets replaced by acetyl group, then N-acetyl glucosamine is formed. What is acetyl group? It is the common name for, acetyl is the common name for ethonyl group. So what is ethonyl group? Ethonyl means two carbon, right? So this is carbon number 1, carbon number 2. Carbon number 1 is normal, which means it is fulfilling rest of its all three valencies with the help of 3 hydrogens. While this second carbon, which is there, fulfills its valency with the help of a carbonyl group, which means a double bond O. Then this group which is formed, this is called, in IUPAC, it is called ethonyl. Why? In common, its common name is acetyl. From this acetyl group only, if this acetyl group is associated with H, then it will form aldehyde, which can be called as ethanol, right? Or we can say acetaldehyde in common language, or in the its common name will be acetaldehyde, while its IUPAC name will be ethanol. While if it will get associated with R, means another methyl group over here, then or ethyl group, it can be anything, CH2OH6, then it will form a ketone. And if it gets associated with OH over here, which we will do it in the next slide in quite detail, if it will get associated with OH over here, then it will be forming a carboxylic acid, and it will be called ethanoic acid in terms of IUPAC naming, while it will be called acetic acid in the according to the common naming. So over here, this group is generally called acetyl group. So this is this acetyl group, this much is the acetyl. So you can see one of the eights over here, in NH2, it is left with just one H, has been replaced by this group, acetyl group over here, two carbon group, in which one of the carbon is associated with a double bond. So this is the acetyl group. So the molecule, the derivative which is formed is really important. We will come across it when we will be doing the polysaccharides in the later videos. Then after this, if once again we will go back to this glucosamine. If in this glucosamine molecule, this is right, this OH over here gets replaced by a very bulky group which is called lactic acid. So what is lactic acid? This is the lactic acid, right? Lactic acid is a 3-carbon molecule where one of the carbons is actually is basically involved in the formation of the functional group carboxylic acid. We will do carboxylic acid in detail in next slide. And one of the carbon is a simple methyl group, while the central carbon is associated with OH generally, but over here this is lactic acid basically, if this would have been associated with H over here. So this is lactic acid, the central carbon associated with OH on one side and H on other side. One of the terminal carbons is actually involved in the carboxylic acid formation, and the third carbon being simple methyl group. So this is the lactic acid. Now this H over here is replaced by this carbon number 3 over here, right? This H will be replaced over here with this carbon number 3. It will get associated with this carbon number 3 by replacing this OH over here. From this OH will be replaced, and over here H will be replaced, and this ether bonding will happen. This is carbon COC, right? COC we have done, that is the ether bonding. So it will get associated in glucosamine through ether bonding at carbon number 3. And the molecule which is formed in such a manner is called muramic acid. That is called muramic acid, right? Simply from here again, this is also the N-acetyl glucosamine, that is also the derivative of glucosamine, which is again the derivative of glucose. So glucosamine is the derivative of glucose, then N-acetyl glucosamine is derivative of glucosamine. Again, muramic acid is also the derivative of glucosamine. Now we need to do the derivative of this N-acetyl glucosamine. In this N-acetyl glucosamine also, when this OH present on carbon number 3 gets replaced by again with lactic acid, then the structure which is formed is called N-acetyl muramic acid. So these are all the basic structures of the derivatives of glucose. These all are the just normal derivatives of glucose, and they are really, really important. We will come across each one of them everywhere during our studies. So if you have a good knowledge of their structures, you will have a friendly acquaintance with all of them. The first is glucose 6 phosphate, in which on the carbon number 6, H of hydroxyl group associated with carbon number 6 gets replaced with the phosphate group. Glucose 6 phosphate is formed. This is also called, the formation of this glucose 6 phosphate is also called activation of glucose or activation of any other monosaccharide, whichever is getting phosphorylated on the carbon number 6. Then if this glucose, in this glucose, OH get on carbon number 2 gets replaced by the amine group, that is NH2, then glucosamine is formed. Now in this glucosamine, if one of the H's of glucosamine group gets replaced by acetyl group, then N-acetyl glucosamine is formed. Now when in this, again in glucosamine only, right? First modification is that OH is replaced by the amine group. Second modification being that the OH group on the third carbon gets replaced by the lactic acid through ether formation. So this R group is representing the lactic acid because it's a quite bulky group, not possible to show over here. So we have represented it over with the R group over here. So this is the muramic acid, and this over here, again, if same thing happens in N-acetyl glucosamine, that this OH on the carbon number 3 gets replaced by lactic acid, then N-acetyl muramic acid is formed. So now let's proceed further. Now we need to discuss the alcohol sugars. Before we discuss the alcohol sugars, I need to mention a fact from organic chemistry. Now, what is that fact? The fact is that whenever any alcohol gets oxidized, right, it forms aldehyde. It forms aldehyde. And whenever any aldehyde gets oxidized, it forms carboxylic acid. Whenever any carboxylic acid gets reduced, then it forms aldehyde, right? And whenever any aldehyde gets reduced, it forms alcohol. It forms alcohol. So these are the two facts that alcohol's oxidation will lead to the formation of any aldehyde, and any aldehyde when gets oxidized, it forms carboxylic acid. So it actually means that the eventual result of alcohol's oxidation can be the formation of carboxylic acid. Okay. Now, as we did in the earlier videos, especially in the video Biochemistry 2 and Biochemistry 3, that what is the general formula of alcohol? It is that whenever any alcohol binds with oxygen through a single bond, then that oxygen, to fulfill its divalency, means it needs to form two bonds, it gets associated, or we can say it gets bonded with hydrogen. So that forms the general form of alcohol. We call this group hydroxyl group, this OH group, as hydroxyl group. So presence of this hydroxyl group actually tells us that this particular molecule is an alcohol. Now, then again, in the video Biochemistry 3, we did what is aldehyde. We did the general formula. It was that whenever there is any alkyl group and that alkyl group is associated with such a carbon in the end, where that carbon is associated with oxygen but through a double bond. Three of the valencies of the carbon are fulfilled. Now, when it fulfills the fourth valency with the help of simply the hydrogen, then an aldehyde is formed. This is the general formula of aldehyde. Now, this carboxylic acid. Now, this is the new addition. I have not introduced this functional group earlier. So what is the general formula? This is same as that there is an alkyl group, the way it was over here. It gets associated with such a carbon which is bonded with oxygen through a double bond, just like over here. But now, three of the valencies of this carbon over here are fulfilled. Now it needs to fulfill the fourth valency. Now for that, it gets associated with oxygen over here through a single bond, right? Through a single bond, it gets associated with oxygen over here. Now this oxygen, further to fulfill its divalency, gets associated with hydrogen. So basically over here there is a carbonyl carbon as well, and there was a hydroxyl group as well. So whenever in any functional group there is carbonyl carbon as well and the hydroxyl group as well, then this general formula of carboxylic acid is formed. Then that molecule is called carboxylic acid. Whenever this alcohol will get oxidized, it will form this, and when this aldehyde will get oxidized, it will form this. So this is the general, and the reverse is also true, that whenever this will get reduced, this will form aldehyde, and when this will get reduced, it will form the alcohol, right? This is the reduction form, and this is the oxidation pathway. We know that the monosaccharides are polyhydroxyaldehydes. It means we are here. Whenever this aldehyde will get reduced, it can form alcohol, and whenever this aldehyde will get oxidized, it will form carboxylic acid. So this fact from the organic chemistry is the basis of the formation of these alcohol sugars, which are mentioned over here. And in the next slide, we are gonna do the acid sugars as well, right? So over here, this funda, that aldehyde, whenever gets reduced, they lead to the formation of alcohol. So this is the basis. So this, first of all, this alcohol over here, which is named as ribitol, is derived from the polyhydroxyaldehyde, or we can say aldose, which we know as ribose. So ribose is basically a 5-carbon compound, right? So it is a 5-carbon, it is basically aldopentose, right? So aldopentose, 5 carbons are there, 1, 2, 3, 4, 5. Now in those 5 carbons, we know that this uppermost carbon is associated with a carbonyl group and hydrogen, just like over here, this, right? This carbon. Now, this is the R form. In the R form, we know that it is polyhydroxyaldehyde. It is not the simple aldehyde, it is polyhydroxyaldehyde. So each of the carbons need to be associated with the hydroxyl group, but on the interstitial carbons, we need to be careful about the orientation of the hydroxyl group, or we can say the configuration of the hydroxyl group. And that configuration comes from the code. And for the code for ribose is 2 3, 4, which means on all the 4 carbons, all the 3 interstitial carbons, hydroxyl group is associated, is present on the right hand side of the structure, and over here it is on the left hand side, right? So you can see over here, this is the ribose sugar from which this alcohol, which is called ribitol, is derived. So what happens, this carbon number 1 over here gets reduced, it gets reduced, and when it gets reduced, then it loses this bond. Instead of bonding with double bond O, it gets bonded with just a single bond O, and that oxygen further gets associated with hydrogen. So another additional hydroxyl group comes into the picture. Now, if we check over here, this carbon is actually is not fulfilling its tetravalency right now. So it to fulfill its tetravalency, it gets associated one more hydrogen. So the resultant is CH2, CH2OH, right? CH2, CH2OH. So in this way, the ribitol is formed. It is a reduction of this polyhydroxyaldehyde. Same as over here with glycerol. Glycerol is derived from glyceraldehyde. In glyceraldehyde, this carbon over here was involved in the formation of carbonyl group, but now it has lost its carbonyl group, and it is this OH group has been formed on it, and this way this polyhydroxy alcohol, basically these all are the polyhydroxy alcohols which are formed. Glycerol is very important derivative because it is the part of lipids as well, so this is really important. And then same is the case with xylitol. It is formed from the, again, pentose sugar, aldopentose xylose. So same things are there over here. So over here, everywhere, this carbon number 1 is getting reduced. The carbonyl group on this carbon number 1 is getting reduced to form this alcohol. This COH is getting converted into OH, right? It is getting associated with OH. Now, I am again and again referring to 2 terms, that is oxidation and reduction. Here, just an introduction. What is oxidation, first of all? Now, oxidation is basically the addition of oxygen, as the name suggests. So wherever there is an addition of oxygen, then you can call that oxidation is taking place. Removal of hydrogen, right? Removal of hydrogen. So whenever you see that the hydrogen has been removed, addition of oxygen may or may not have taken place. But if even if the addition of oxygen is not taking place, but the hydrogens are being removed from any atom, then also that it means that atom has been oxidized. And similarly, then there may be the case where there is no addition of oxygen and there is no removal of hydrogen, but still the oxidation can happen when there is a removal of electrons. Let's go to the reduction. Reduction, because oxidation is addition of oxygen, so reduction is the opposite of it. So it is the removal of oxygen, basically. Oxidation is addition of oxygen, so reduction is removal of oxygen from any atom. Similarly, it may or may not happen, right? Removal of oxygen may or may not happen. Then in that case, there may be the addition of hydrogen to any group. So if there are additional hydrogens are added to any group, then also this can be the reduction. Now, sometimes there is no removal of oxygen, there is no addition of evident addition of hydrogens, but there is just simply the addition of electrons. We can see that some electrons have been added to this group, right? So in that case, we say that again the reduction has taken place. Now over here, with what is the condition? So I'm saying that over here this aldehyde is getting reduced. So aldehydic group, when this carbon was actually an aldehydic group, I am just taking the case of this one, this carbon, right? When it was aldehydic group, so what it was actually, it was this CH and just this O, and there was this whole of this we can consider as R, right? So this is the thing. This was the thing till this. So over here we can see there is oxygen and there is one hydrogen, right? So even if we count over here after reduction also the carbon is associated with one oxygen. So one thing is very sure, there is no addition or removal of oxygen over here. Then over here, there was just one hydrogen. Now over here, it is associated with two hydrogens, right? So there is addition of one hydrogen, extra hydrogen atom over here. This we can just see by in a very simplified manner, right? So so there is the addition of hydrogen. Addition of hydrogen means reduction. So this proves that here this molecule of ribose is actually undergoing the process of reduction to form this alcohol, which is called ribitol. So this happens, same thing happens with glycerol, and same thing happens with xylitol, or for that matter, any other every other monosaccharide is capable of undergoing such kind of reduction to form their respective alcohol derivatives. Now, as we have done this, we need to step on to the next slide where we need to do the acid derivatives, right? So in the acid derivatives, we know that it will be the oxidation. Acid derivatives of acid derivative of these polyhydroxyaldehydes will be formed by the process of here. The alcohol derivatives were formed by the process of um, reduction, right? But the acid derivatives will be formed by the process of oxidation. Over here, the same thing is applied, that whenever aldehyde, any aldehyde gets oxidized, or any alcohol for that matter gets oxidized, it ultimately leads to the formation of carboxylic acid, right? So over here, this carbon is associated with an alcoholic group like this hydroxyl group, and this hydrogen group, if we just talk about the carbon number one, right? So whenever this carbon number one gets oxidized and it forms the carboxylic acid, after getting oxidized, then the acids which are formed are called aldonic acids. Their names are called aldonic acids, and they are named as that from whichever monosaccharide they have been derived. For example, this has been derived from glucose. It has been derived from glucose. That's why it will be named as gluconic acid. Gluconic acid. Now, what is the catch? That these acids, like acid is when this is associated with like this group, right? When there is C double bond O, and over here it is associated with oxygen, which is further associated with hydrogen, then you will call this group as acidic group. But what happens that at the physiological pH of 7, this OH gets dissociated from this oxygen, right, in such a way that the negative charge is, this is again a heterolytic cleavage, then this oxygen gets the negative charge and the H positive gets removed from here. So when this happens, then you call such groups, rather than although the nature is acidic only, their name adds with this word ate. So over here, gluconic acid can be called gluconate whenever it just, and it is a reversible process. Once the pH will go down, then it can actually become again gluconic acid, right? It just needs the excess of hydrogen. So gluconic acid is similar to gluconate. So over here the name gluconate is written, actually it is gluconic acid. All the monosaccharides which get oxidized at carbon number 1 form the aldonic acids, and they are named in such a way. If it would have been galactose rather than glucose, which is getting oxidized at its carbon number 1, then it would have been named as gluconic acid, right? Now, this is the first option, that the carbon number 1 gets oxidized to form the carboxylic acid, this aldonic acids. But when this carbon number 6, this is the carbon number 6, right? Over here, the carbon number 6 is untouched because carbon number 1 has been oxidized. Now, over here, this carbon number 6 over here, when this gets oxidized, when the carbon number 6 gets oxidized to carboxylic acids, then the acids which are the acidic sugars which are formed are called uronic acids. Their names are actually enough for us to know that which carbon has been oxidized. So over here, this carbon number 6 is getting oxidized. That's why this is called, its name is uronic acid. But again, the thing is same, that this carbon also loses its hydrogen at the physiological pH. That's why they are called uranates. So glucouronate it is called, because it is derived from glucose. If it would have been derived from mannose, then similarly mannouronate. And if it would have been derived from galactose, then it could have been named as galactouronate. So uranates are formed. Now, if you look over here, this structure where the carbon number 1 was oxidized, you can see that right now, this is the linear structure. Now, this linear structure has all the components which we could see in the case where we were doing the cyclization of polyhydroxyaldehydes and ketones, right? Their intramolecular nucleophilic attack could happen just because both the carbonyl group and hydroxyl group were present in the same molecule, right? So over here also, if you would say this is the linear molecule right now, and there are so many hydroxyls, and this is the carbon number 1 with a carbonyl group over here once again. So when this happens, then again the nucleophilic attack of this oxygen takes place on this, and the cyclization takes place. But this time, because this time it is not the aldehydic group on which it is attacking, it is the carboxylic group over here. So this time a derivative of monosaccharide is formed, which is called d-glucodelta lactone. So these kinds of derivatives can also be formed from the linear acidic sugars, right? So linear acidic sugars get cyclized to form these lactone structures. Now, apart from this, here we could see that either the carbon number 1 is being modified, or carbon number 6 is getting modified, or the groups present on carbon number 2 were getting modified. We did the amino sugars. Maximum when we were doing the muramic acid, then over there, the 2 of the carbons were getting modified, right? But over here, if you see this kind of derivative, which is N-acetyl-nuramic acid, then you will notice that over here, the carbon number 1 is getting modified, right? To this carbon number 1, additional carbon is getting associated, which is in the form of carboxylic acid. This is the first modification. Then carbon number 4 is also getting associated with this N-acetyl. Then this group, carbon number 5, is also getting associated with this kind of 3-carbon polyhydroxy alcohol, right? So there are the 3 major groups are getting, newly additional groups are getting added to the same molecule. And so this complex molecule is called salicylic acid in general, or it is called N-acetylnuramic acid. This is one of the most complex derivatives of the monosaccharides. Now, let's move to the last derivatives of the monosaccharides, which are called deoxy sugars. This is deoxyribose, which is derived from ribose sugar. We know that ribose is aldopentose and its code is 234, right? Its code is 234. 234. So it means on carbon number 2 also, there needs to be a hydroxyl group which is on the right hand side, right? So carbon number 2 also, on carbon number 3 also there needs to be hydroxyl group on the right hand side, and carbon number 4 also there needs to be hydroxyl group. But because this is the cyclic structure, so we will not consider the carbon number 4. So you can see that on carbon number uh, 3, there is hydroxyl group which is directing downwards as it should be. But on carbon number 2, there should be hydroxyl group. But instead of that hydroxyl group has been replaced by just hydrogen. So hydroxyl group had one oxygen in it. This hydrogen is without any oxygen. So that's why it means one oxygen atom has been removed from whole of this ribose sugar. That's why this is no more known as ribose, but deoxyribose. And this deoxyribose is very, very important component of our DNA. While ribose in its original form is the component of RNA. Then we come to these derivatives, which are actually the derivatives of the rare enantiomers. When we were doing the enantiomers and the apimers, in the video Biochemistry 2, we discussed that these enantiomers are there. Whenever there are the enantiomers are of two types, D and the L forms, right? D and the L forms. D forms are when the chiral carbon farthest from the carbonyl group has the hydroxyl group directed towards the right hand side, on the right hand side of the carbon skeleton, then it is D form. And when the hydroxyl group on the chiral carbon farthest from the carbonyl group is towards the left, then that group or that enantiomer will be called the L form, right? Most of the monosaccharides in our nature are in the D form. Wherever they are present in the L form, they generally exist in form of their derivatives only. So for example, this derivative, which is L-rhamnose. So what is there? It is the derivative of mannose. Mannose is a 6-carbon compound. So and the code for mannose was 45, which means on carbon number 4 and 5, hydroxyl group needs to be on the right hand side, right? On carbon number 4 and 5, just on carbon number 4 and 5, it should be on the right hand side. But this is true for D-mannose. When I want to talk about L-mannose, code will remain same, but for L-mannose, this code will depict, or it will tell us the carbon positions on which OH groups are on the left hand side. The code is same. Same code for the D form will tell us the positions, the carbon positions on which the hydroxyl group is present on the right hand side of the carbon skeleton. But in the L form, same code will tell us the positions, the carbon positions on which the hydroxyl groups are present on the left hand side of the skeleton. So right now, as we are dealing with L-mannose, not the D-mannose, so these hydroxyl groups on carbon number 4 and 5 will be present on the left hand side, right? It means on carbon number 2 and 3, hydroxyl groups will be present on the right hand side. This is totally opposite of the D form, right? And that is why this is enantiomer. Enantiomer is the mirror image. Whatever was depicting the things to be on the right, same thing is depicting the things things to be present on the left right now. So enantiomers are also called, that's why the enantiomers are also called the mirror images. They are the mirror images. Molecules need to be the mirror images of each other. So over here, this is the case. And the carbon number one, because this is aldohexose, this will be associated. So this is H, and this is H over here, this H, a chiral carbon, which is associated with two hydrogens and one hydroxyl group. This is the basic structure of L-mannose. Now, when this will get cyclized, then the rule about cyclization will remain same. So whatever is on the left-hand side of the chain, that will be directed upwards on the ring. And whatever is on the right-hand side of the chain will be directing downwards on the ring. So this basic rule will remain same. If you will go by this rule, you will see that we need to start from carbon number 2. On the carbon number 2, OH is directing downwards. On carbon number 3, OH is directing downwards. On carbon number 4, OH is directing upwards. Carbon number 5 will actually get involved in the ring formation, so no problem. And now this CH2OH, CH2OH, over here, let me write this initially. This is directing downwards over here. If it would have been the D form, it would have been directing upwards. Right now, it is directing downwards. This I actually mentioned earlier as well. If it, in all the D forms, this sixth carbon or whatever the carbon is overhang, these are the overhang carbons. These overhang carbons in the D forms, they are directed upwards, and in the L forms, they are always directed downwards. This is also one of the ways to see that whatever the cyclic structure is there in front of us, whether that is the D form or the L form. So now this is the original thing. Now what is happening? This OH is being removed from here, right? Just the way from here it was, OH was removed. Again, this OH is removed and it is replaced by additional H over here, right? Additional H over here, due to which this CH3 is formed. So again, a group containing oxygen has been removed, and which means oxygen has been removed. So that's why this is also called, this is also a deoxy sugar, right? This is deoxymannose. Just it was deoxyribose. Now there is a general name for deoxymallmannose, which is rhamnose. Now, we need to discuss about fucose. Fucose is the derivative of galactose, but which galactose? Not D-galactose, but L-galactose. Now, code for galactose is 25. So this code 25 is telling us what? That hydroxyl groups are present on the right-hand side of the carbon skeleton? No. Here the same code is telling us that on carbon number 2 and 5, hydroxyl groups are present on the left-hand side of the carbon skeleton. Left-hand side of the carbon skeleton. Let's make the changes in this molecule only on 2 and 5, right? So let's make the changes over here on this skeleton only. On 2 and 5, they should be present on the left hand side, so over here and over here, right? In all other positions, it means OH on the right hand side. So that is the L-galactose basically. So over here, if you will check, everything is fine over, and when we are converting it into the cyclic structure, the rules don't change. Whatever is there on the left-hand side of the carbon skeleton, that should be pointing upwards on the chain, on the ring, and whatever is on the left-hand side of the carbon skeleton, that should be directing downwards on the ring, right? So you can see we need to start from carbon number 2, on carbon number 2, you can see that OH is directing upwards. Yes, it should be directing upwards. Then it is directing downwards. Then it should be directing downwards. Then carbon number 5 doesn't matter because it actually loses its hydroxyl group during the process of cyclization. But this CH2OH, which in normal D form would have been just pointing upwards, here it is pointing downwards, and it has been, its hydroxyl has been taken from it and it has been replaced by one extra H, and that's why this is deoxygalactose, commonly carbofucus. One more point. What is that point? Over here, check over here. Here hydroxyl is a group on carbon number 1 is pointing upwards. Here also the hydroxyl group on carbon number 1 is pointing upwards. Till now what we have been doing, that whenever the hydroxyl group points upwards, then we name that stereoisomer of that cyclic monosaccharide as beta. But here the point to be noted is that that is true only for the cyclic structures of D monosaccharides. If we are dealing with the L monosaccharides, then that situation is also reversed, which means that in the case of L monosaccharides, which are actually very rare, so don't get confused. It is just an additional point which you need to just keep in mind, right? Actually, in case of D and L forms, whatever is true for D form, reverse of it is true for L form. That is the only difference. If in the D forms we are doing that beta isomer is formed when OH points upwards, for L form, beta isomer is formed when OH group points downwards. Whenever it points upwards for the L forms, it is the alpha isomer. So you need to keep the things related to D forms in your mind because they are the monosaccharides which are found in nature, right? L forms are rare. So don't get confused with the L forms. Yes, you should be knowing that everything opposite of D form is true for the L form, but still, just you need to concentrate on the D forms. So here also the same thing is happening. This are otherwise if it would have been D form D isomer, then this position would have been, we would have called it beta isomer, but over here this is alpha isomer, right? Here also it is alpha, it is not beta because it is the L form of the monosaccharides. So with this we are ending our derivatives. If you have any question, any confusion, you can always ask the questions in the comment sections. I will be really happy to address all your doubts.
So now we are winding up with this topic, the structure of monosaccharides. In the next video, we will do some analytical questions related to this topic. And thereafter, we will proceed with the structure of oligosaccharides and polysaccharides. So till the next video, goodbye and take good care of yourself.